We are starting with a series of experiments which perform to prove DNA is a genetic material. So the first step or the first experiment is the Griffith experiment, which performed in the year 1928. The Griffith experiment was performed in the year 1928. After Griffith experiment, because Griffith experiment is completely failed to prove whether DNA is a genetic material or not. So after 16 years of Griffith experiment, that means in the year 1944, after 16 years of the first experiment, the second experiment to prove whether DNA is genetic material or not was carried out by three scientists. They are Avery-MacLeod-Mc. So the second experiment which tried to prove DNA is genetic material or not is carried out by three scientists, Avery-MacLeod and Mc. So they carried out this experiment in the year 1944. These three scientists also performed or pretty same like Griffith experiment, but with few additional things. Here the few additional things are nothing but, apart from carrying the case 4 experiment, they have used even enzymes, different enzymes at different times. So, according to case 4 in Griffith experiment, that Griffith has used two strains. First one is R2 lime, which has been combined with S3. But here S3 is not virulent strain, but here the virulence is destroyed by exposing the strain to heating. So the strain is called heat-killed smooth strain 3. Now the strain is called heat-killed smooth strain 3. So we use two combinations, R2 lime and heat-killed S3. As like as same in case of case 4 of Griffith experiment, these three scientists also performed with the same strains. So when they injected into mice, we can expect the result here. As we know, R2 is virulent in nature. Whereas heat-killed S3 is virulent in nature. So both these strains are virulent. That means they don't have a capability to cause disease. So fine, when we are injecting these two strains to the mice, as we know, it won't cause any disease to the mice. So we can expect the result, the mice definitely should survive. But here the results are wouldn't reach the expectations. Here the mice died. This is a surprising part of whether Griffiths or Avery. This is a surprising part that their expectations wouldn't reach the result. So here the result is not matched with the expectations of Griffith or Avery-MacLeod. So what happened in the next case is these three scientists took isolated tissue from the dead mice. They isolated the tissue from the dead mice. When they isolated and studied, they found S3 strain in the tissue. How does it possible? As they used R2 lime and heat-killed S3. Both are virulent, don't have a capability to cause disease. But after the death of the mice, when they isolated the tissue from the dead mice, they surprisingly found that the tissue of the dead mice contained S3 lime. That means smooth strain 3 live condition. That means it is having a capability to cause pneumonia in the mice. It has a capability to cause pneumonia in the mice. So the reason behind this case 4, why the mice got died in case 4 is because this heat-killed S3 has been converted into normal S3, nothing but virulent S3. So that's the reason why the mice died in the 4th case. So here the heat-killed S3 cannot convert into live S3. There is no possibility that heat-killed S3 can able to convert into live S3. So there is another possibility. They thought that there is another possibility of transformation here. Here transformation is nothing but change in the form. That means S3 may convert it into R2 or R2 may convert it into S3. This changing in the form is nothing but transformation. So here what they expected is either the heat-killed S3 may have converted into normal S3, virulent S3, or else the second possibility is R2 may converted into S3. So the second possibility is R2 may have converted into S3. Here the heat-killed S3 do not able to convert it into live S3. The heat-killed S3 do not able to convert into live S3. But there is a chance of R2 may have converted into S3. That means here some molecule in heat-killed S3 stimulated R2 to convert into S3. Here some molecule in the heat-killed S3 may have stimulated the R2 to convert into S2. What is the major difference between R3 and S2? The only difference, not major even, the only difference between R2 and S3 is like it is smooth strain. Smooth strain is nothing but around this bacteria there is a slimy capsule. Around this bacteria there is a slimy capsule, where this capsule is the reason for causing pathogenicity. So whereas coming to the rough strain, where the name itself indicates it is rough in structure because of the absence of capsule around it. Because of the absence of capsule around it, it appears like granule-like structure. That means it appears rough in nature. As we know, here capsule is not there. That means they are non-pathogens. They are non-pathogens. Or it won't have an ability to cause the disease. But here, when R2 has converted into S3, that means some molecule here in S3 stimulated the R2 to convert into S3. That means here the R2 developed a capsule around it. When it develops a capsule around it, what happens? It becomes smooth. That means here R2 is live. So when it develops a capsule around it, it becomes S live. That means smooth strain live. Immediately what happens when it turns into smooth strain live? Immediately it can able to cause disease. That means it turns into virulent in nature. So this is the reason for the death of mice in the case 4. Instead of surviving of the mice, the mice got died because some molecule in the heat-killed S3, when combined with the R2, the molecule in heat-killed S3 has converted the R2 live into S3 live by synthesizing a capsule around it. So when the capsule got synthesized, it becomes virulent in nature. At the same time, cause the mice to die. So they performed the same experiment with the help of enzymes. So they use enzymes to know the molecule, which molecule it is. So the 3 scientists have used 3 different enzymes: proteases, transformation. These proteins are not responsible for transformation. Transformation is changing in the form. That means proteins are not responsible for converting R2 line into S3. So fine, here proteins got clarified. Next macromolecule is RNases, which destroy the RNA, which is also a genetic material only in some viruses, whereas it acts as a base for synthesis of proteins in other animals. Well, when RNase enzyme is used, it destroyed the RNA. When carried out the same experiment, they do got the same result in here also. Again the mice done. So when the result is matching with the previous thing, that means this molecule is not responsible for the transformation capacity. So RNA is not responsible for converting R2 into S3. So fine. In the third case, when they use DNA, which got destroyed the macromolecule DNA. When the DNA got destroyed, they got the result like this. The mice survived, or the mice line. The mice survived. Here the result is different. At the same time the molecule is also different. The result is again different here. At the same time molecule is different. That means this molecule, nothing but DNA is responsible for the transformation capacity. DNA is responsible for converting R2 line into S3. So the some molecule in heat killed S3 is nothing but the DNA present in the heat killed S3 stimulated the R2 to convert into S3. So when it immediately converts into S3, the mice died there. But here when the DNA got destroyed, so nothing is there to harm the animal here. So hence the mice got survived here. So after this experiment, the three scientists come into conclusion that this DNA acts as genetic material. But to carry out this experiment, the Griffith experiment acts as or laid as a base to carry out the experiment 2, that is which is carried out by the three scientists Avery-MacLeod and McCarty. So the Griffith experiment is the first experiment which scientists gave some clue to carry out the experiment 2. So this experiment which is carried like Avery-MacLeod-McCarty experiment, which finally proved that DNA as a genetic material.